molecular mechanisms. Thank you, Marom.
Thank you, Sven. Thank you, Leo. Thank you to the organizers for the opportunity to speak here today. They also asked me to facilitate a consensus paper by individuals really focusing on the cellular mechanisms of non-invasive brain stimulation using animal models. And so what you'll be hearing today is an overview of some of the themes that came out of that discussion towards this consensus paper. So it's useful to start with what really is the goal of animal research around non-invasive brain stimulation, be it TMS or be it tDCS. And I want to emphasize here, this is the goal of translational animal research. So not just asking questions for their own sake, for the academic sake, but trying to inform clinical practice. And so the goal of these models is to inform clinical paradigms, is to provide suggestions to clinicians, to neuroscientists who are using non-invasive brain stimulation on how they can apply those techniques more effectively, leading to more specific outcomes. And you've just heard several talks on why this is so key. Now, we have current flow models of the head. These are those pretty pictures that you saw in John Rothwell's talk. Models that represent how current flows through the head during non-invasive brain stimulation. This is only one piece of the puzzle, though, because knowing where the current is flowing does not tell you what it does to the brain. That's only the input. And we've also heard there's quite a lot of ongoing human neurophysiology and also clinical trials trying to address these very same questions. But I think one theme that's really emerging is that the parameter space is huge. 11 hertz, 11.1 hertz, 11.1 hertz with 50% duty cycle. It's almost intractable, especially once you start talking about the fact that the behavior itself will affect what it do. And so for this reason, animal models may be able to facilitate this process. And an important idea, if your goal is only translational, is that we're okay with empirical models. While you might think, well, animal research must be really obsessed with mechanisms, if we can develop equations, if we can develop predictions that facilitate this process, but we don't quite know, is it glia, is it interneuron, but we can make the right predictions, then we've achieved at least this very limited aim. And indeed, for example, if you look at seminal work on neurophysiology, often they don't have all the answers, but they're able to make models that are very effective in making predictions. And with that sort of theme in mind, I think it's also important to think about not one model for one experiment, but trying to think about a framework, trying to think about a way where we can pool our resources to develop something that we share, and that's something that is robust to a variety of different techniques. And I'll also be touching a little bit on biomarkers. In a sense, every animal study is measuring a biomarker. And I want to talk about what's the most effective way to translate that to the clinical setting, while being very cognizant, right, of the limitations of animal models. So a lot of non-invasive brain stimulation is predicated on themes related to the sliding scale notion of the brain, and it's not endogenous to brain stimulation, where if you have brain function or brain dysfunction, you're sort of moving on the slide, right? So if you are depressed, you move down the slide in the prefrontal cortex. And if you have epilepsy, you moved up the slide somewhere else. And again, this is something that derives, it has a long history of where this sort of thought process comes from, including from imaging, where things light up, right? And even if you're willing to accept that non-invasive brain stimulation sort of produces this sort of monotonic change in function, which we've heard it doesn't, this idea obviously is very limited. So this is the sliding scale idea. And so non-invasive brain stimulation has either inhibitory forms or excitatory forms, and at the most basic level, they're assumed to sort of move this sliding scale one way or another. So if you have a disease that's associated with hypoactivity, you will use an excitatory form of non-invasive brain stimulation to move the sliding scale. And I want to touch on, and there's certainly a lot of data to support this idea, but it's also obviously not as sophisticated as it could be. And so it's really, for me, about making non-invasive brain stimulation more specific. So we already know that it works for everything, right? That has certainly been established. You can apply it for any disease, you can apply it for any cognitive function, and it will produce enhancement. But if that is the case, where does the specificity come from? Why does it work for depression in Brazil, and it's working for pain in Boston, and it's working for mathematical skills in England, and so on? I think the specificity really gets into the heart of trying to make this rational. And there's two basic sources. One is anatomical targeting, anatomical specificity. And this underpins the use of, for example, figure of 8 coils to be more specific than rings. It's about getting the current to specific brain regions of interest that are involved in those tasks. But there's another form of specificity that I think is equally important and that I think the animal studies can start to address. And this is functional targeting. It's very well established, we've heard about this, that often non-invasive brain stimulation is applied as adjunct to some form of training or some form of other therapy. So for example, if you have a stroke and you're starting to restore function in the hand, you don't just apply tDCS on its own, you apply it in conjunction with manual or robotic therapy. So, and the idea is that perhaps this combination in itself not only facilitates efficacy, but it also facilitates specificity. And so I'll really be spending the talk talking about where might that specificity come from. Just touching back a little bit on the anatomical, so we've known now for maybe about half a decade that using arrays, almost in the form of an inverse EEG, models predict that you can focalize current to the brain, and there's been several trials using these forms of techniques to try to be focal with electrical stimulation, perhaps it's approaching the focality that you might get with transcranial magnetic stimulation. And it turns out that this problem is a rather closed problem, meaning that if you have a brain target region and you have whatever constraints you want, upper limits on current, how many electrodes you want to use, safety concerns, you can mathematically produce a best montage. A computer program will spit out the best montage for that purpose. But what is best will depend very much on what you want to achieve. And a key factor to consider is intensity versus focality. So if you just want to get the maximum amount of intensity in one part of the brain, you just want to blast it, right? You're likely just going to put electrodes across the head. While the best montage for focality would be rather different. But even given the progress that has been made in focality, both for now with tDCS and certainly for TMS, I want to talk about the limits of anatomical targeting that lead to functional targeting. So let's assume we have now a specific gyri, or even a crown of a gyri, and that gyri has different cells inside of it that are responsible for different functions. And let's say the green ones are the ones that do math, and the gray ones are responsible for memory. We want to touch the green ones. Now these are literally overlaid, right? They're overlaid at the micron level. So no matter what we do with stimulation, we will influence both of them. There's a limit to anatomical targeting where you will inevitably hit a part of the brain. If you assume that part of the brain does more than one thing, you will affect these multiple networks. Where functional targeting comes in is the notion that if the network that you are interested in, the green one, is somehow preferentially active, maybe because of disease, or maybe because you prime it through training, now the stimulation might preferentially influence just that network. And so this idea is actually separate from anatomical targeting. In fact, it allows you, if you can do this very effectively, anatomical targeting becomes much less important. You can actually stimulate the whole brain. So these are two separate ideas. In the case of anatomical targeting, I feel like you very often come back to the sliding scale idea. If you just think about anatomical, I think it's a little bit difficult to move around it. When we talk about functional targeting, you can start talking about activity-dependent changes. And I think this opens up a door to increased sophistication. And this is really where a lot of, I think, the animal studies that we're seeing right now are pushing. They're pushing specifically towards developing a quantitative description of that, right? A quantitative description that could be used to make predictions. Now, we're not there yet, but I want to show you, how far we've come. And so I want to focus specifically on tDCS to make some of these points. And we've certainly heard a great introduction to it. Now, at the single cell level, what tDCS does is it polarizes neurons. And there's been a tremendous amount of animal research telling us how much. It turns out it's not very much at all. It's about 0.1 millivolt per volt per meter, which translates to less than 0.1 millivolt per milliamp applied. It's a very tiny polarization, and several groups have confirmed this across animal systems. Now, what's interesting is that this polarization is additive on top of whatever's going on. And so what's illustrated here is this notion of this additivity. So if the average membrane potential is at a certain level, we will polarize on top of that. But if that average membrane potential is at a higher level, let's say due to more ongoing synaptic activity, we will increase that polarization. And this additivity, maybe it seems trivial, but it's actually rather important from this notion of functional selectivity. It means that those neurons that are active are going to be pushed to a different level than those neurons that are not active. Another fundamental result from animal studies is the notion of firing rate. And I think this is really, these studies in fact gave us some of the hints that prompted the early clinical neurophysiology. And one of the most basic findings with animal studies of direct current stimulation is that you can change firing rate. That a nodal stimulation increases firing rate, and that cathodal stimulation decreases firing rate. And this has been replicated across many systems, many animal models. And again, it formed a very important part, I think, of the origins of clinical tDCS, of the origins of applying long duration stimulation to produce lasting effects. That was first observed in animal models. It's sort of the original LTP, in fact. And the change is somewhat linear within stimulation that you apply. So the more stimulation you apply, the more DC current you apply, the more change in firing you get. And so in this sense, it's once again additive. So if a neuron is firing at 10 hertz, you will bring it to 11 hertz. But if it's firing at 20 hertz, you will now bring it to 21 hertz, a new level. And so you have this notion of additivity. On top of that, there's work showing that you can actually get a increased sensitivity to an active system. So meaning that if the system is active, the level of the firing rate, relative firing rate change will be higher for that active system. It's not exactly multiplicative, but it certainly leverages this idea that a more active system will have a bigger change. A whole other class of animal studies has focused looking at the effect of tDCS on synaptic efficacy. And so there's animal versions of the TMS, tDCS paradigm, where you use a targeted pulse to trigger a excitatory postsynaptic potential that can be measured intracellularly or extracellularly, and you apply a field on top of that. And this issue has been looked at for decades, and it has been shown that low-intensity direct current can make these responses bigger or smaller. So that means that even in the absence of firing, the strength of synapses, the synaptic efficacy, can be dialed up or down. And there's been exhaustive work looking at this across the hippocampus, across the cortex, really peeling away individual fiber bundles in a way that would be impossible clinically. But the consensus is basically that a version of a nodal will increase synaptic efficacy, while a version of cathodal will decrease synaptic efficacy, consistent with the sliding scale idea. Now, where it gets interesting is a very simple permutation of this idea, where instead of applying a single pulse, you apply a train of pulses. And that's the experiment you're seeing here. This is in the cortical slice. These are excitatory postsynaptic potentials recorded with a field electrode, and we're applying a train. Boom, 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 boom. And then at some point, the DC field is turned on. And you can see in that red area where the DC field is on, which is an excitatory field, not only does the first EPSP get bigger, they're all bigger. Now maybe this isn't a huge shock in itself, but for people who study long-term potentiation in animal models, they're aware that the ability to sustain a higher level of ongoing activity is what promotes LTP. Anything that you can do, let's say pharmacologically, to allow your neurons to sustain a higher level of EPSPs, that's exactly what triggers long-term potentiation. And what's interesting about this idea is that presumably this would happen at the synapse that is being driven. Right? So a synapse that's not being driven would not undergo this particular benefit, and so it lends itself to functional specificity. There's specific examples on synaptic plasticity in the literature, including by Fritsch and colleagues. So Fritsch showed that if you have a pattern of activity that does not produce in itself plasticity, you can now apply tDCS to make that promote plasticity. So you're changing something that is non-plastic, a level of activity that is non-plastic, to a level of activity that is plastic. And it's very specific to having a certain type of activity ongoing. Others have shown that if you have ongoing plasticity, you can boost it. So in this case, they used tetanic stimulation, a classic protocol, to produce plasticity, and then they showed that applying direct current on top of it gave you more plasticity. So a similar theme, and in both cases, the notion is that if you have a non-active synapse, or non-active in the right way, if you apply tDCS to the system, you will not produce plasticity. And so this certainly lends itself to this notion of functional targeting. But I put a question mark here because to show that, you actually have to probe another pathway to show that that pathway did not undergo plasticity. But the substrate is there. I want to touch a little bit more on this notion of synaptic efficacy and the sliding scale. It is certainly more sophisticated than that. There have been several groups which have been looking at the effects of direct current on synaptic function in active systems, not passive. So if you just take a piece of tissue out of the brain, it's absolutely quiescent. You need to either drive it up or go to an in vivo system. And what they have found that the actual effect of direct current on synaptic function in an active system is not so simple at all. And more often than not, when you apply, for example, an excitatory stimulus, you not only boost excitatory synaptic function, you simultaneously boost inhibitory excitatory function, and you change the dynamics of the system. And it's interesting that a bunch of groups working independently have converged to very, very similar ideas about this notion of how direct current stimulation affects dynamical systems. And the overarching theme is you cannot explain what the stimulation will do without understanding the state of the system, which really mirrors a lot of what you heard in the previous talks. But that also lends itself to functional selectivity. It means that a system exhibiting one kind of behavior will respond in a certain way to tDCS, let's say an oscillating system, while another system will respond differently, even though they're both exposed to tDCS. So this is sort of the high-level picture of this idea, trying to get at where functional selectivity might come from. I talked about firing rate, where a higher level of firing activity may make you more susceptible to tDCS. I talked about synaptic efficacy, where again, an active system, a actively oscillating system, may respond very differently than a quiescent system to DCS. So the DC is sort of picking out those systems. And we certainly have data showing that DC is preferential as far as inducing plasticity to active systems. So all this gives us the potential for functional selectivity, though we haven't quite shown it. And though I'm not going to be focusing on high intensity stimulation, be it DC or TMS, I think it's interesting to consider that, for example, things like TMS, this actually produces firing rate, right? TMS protocols in themselves can produce plasticity, these trains of pulses. And so I think any sort of functional selectivity that derives from these protocols would be something a bit secondary to the triggering of firing itself. But a lot of the same ideas I just showed you would actually map onto TMS as well. As far as the actual mechanisms, and so I've sort of taken the mechanisms here and I've crossed it out and I've written biomarkers. So there's been a lot of work in animal studies looking at what are the biomarkers, what are the marks that tell us whether tDCS will be effective or not. What does tDCS actually change? And this is a growing body of literature. Maybe another way to think about it is this: the brain is really complex. And in the short term, we're probably not going to figure out how the brain works, and we're probably not going to figure out how disease works. I think non-invasive brain stimulation is also rather complex. And so if we want to be a bit realistic, maybe humble, and say we would like to make this translational jump within the next few years, not in the next 100 years, maybe the approach is a little bit different. And rather than just trying to characterize every possible mechanism, what we need to fish out are the biomarkers, the things that predict what tDCS will do. And we need to identify how those, or tDCS or TMS, and we need to identify how those biomarkers respond to stimulation in a way that is hopefully translatable. And I certainly don't want to diminish the importance of mechanism, the importance of drilling down at these levels, but I think at some point we have to come back to this higher level conception. And so this cartoon starts to address how you might develop a framework around that. So you have on the x-axis a certain level of afferent activity, a certain level of neuronal activity. And this will map without stimulation under some form of plasticity. So it's a bit of a cartoon, but it's basically saying, all right, when you have more activity, you might have LTP. When you have less activity, you might have LTD. And certainly you can think of stimulation as moving you along the x-axis. But alternatively, you can think of neuromodulation as changing the actual map from activity to plasticity. So for example, in the excitatory case, you might have an activity pattern that previously did not lead to excitatory plasticity, but now it does. And this framework is certainly not new. This is something that derives itself from decades of animal work and computational studies on synaptic plasticity. We're simply borrowing them for our purposes. And you can be very quantitative once you start developing these types of frameworks, essentially writing equations like plasticity is a function of activity time and something something. And this XYZ, be it BNDF, or interstellar calcium concentration, these are the things we want to get at. These are the things that we want to determine how are they a function of non-invasive brain stimulation. And if we do that, I think we can start to develop something very broad and hopefully something very useful. This kind of thinking certainly takes into account activity-dependent functional plasticity. It's certainly by nature quantitative, and it could be tuned. So these XYZ parameters may be different for different parts of the brain. They may be also very different for different types of tasks, but as long as we know how to map that, we can generalize this form of model. And again, all we need to know is how XYZ and activity change. So I do want to emphasize that there's two things going on here. One is that the non-invasive brain stimulation is changing the activity directly, for example, changing firing. But it's also changing the substrate for plasticity in a way that may be independent of firing. So again, it's making the synapse somehow different, so that a pattern before that produced one form of plasticity now will produce another form of plasticity. And so these are two separate ideas that are going concurrently. A simple computational model that starts to address this notion of sort of hypothesis-driven design. So here we modeled two populations of neurons. There was a presynaptic population, there was a postsynaptic population. And the presynaptic population, we made them fire at a higher rate. We made them fire at 100 hertz. While the the green population was made to fire at 100 hertz, the red population was made to fire at 20 hertz. And this system, this model, had a very basic way to represent when plasticity would happen. And we were able to show in this very reduced model that when you apply direct current to the entire system, so we applied direct current stimulation to both the green and the red, we promoted plasticity specifically in the green because they were more active. And again, this is more of a conceptual idea, but there's certainly other groups very much drilling down to develop these rather precise computational models. And I think that functional specificity is something that cannot be ignored. And so this is sort of the final point. So what makes non-invasive brain stimulation specific? And I think precisely because we're using it for so many things, this notion of specificity has to be considered in order to make this process rational. And anatomical targeting is essential, and a huge amount of work has been put in, certainly in the TMS space, and now more so in the tDCS space, to increase the targeting that you get with that. And computational models are very important parts of that, and these are ready to go, and they're ready to be used. But if you only rely on this, I think you're a bit stuck on the sliding scale notion. Functional targeting is something else. Functional targeting has to do with activity selectivity. It has to do with the right way, the right timing to pair stimulation with some sort of training in order to specifically target that kind of training. And this is a different class of models. These would be cellular level models, but I'm hoping that if these models are quantitative and if they're properly parameterized, they can produce predictions that will increase the both efficacy and specificity of non-invasive brain stimulation. So thank you for your attention.
Thank you, Marom. We have time for questions. Patrick.
Let's assume you get a model out where you can predict in the premotor cortex in two subsets of cortical neurons, you get this and that effect. How does this translate to a systems effect in the human brain and to therapeutic efficiency?
So it's a very good point. How do we go from changes in synaptic plasticity to behavioral changes? And so one of the assumptions with this paradigm is that, for example, with functional targeting, is that if you understand how to boost functional targeting, if you understand how best to pair plasticity with task, that will translate to better behavioral outcomes. But it is an assumption. There is a bit of a gap there. I don't know if I'm not addressing your question.
I would just say you're targeting the wrong resolution because I think it should be at a region level because that's where you basically, I think you consider, I would just suggest that this is interesting and important, but you should do this at a level of cortical brain systems or kind of at a brain systems level in order to go to behavior.
Oh, I understand your point. Yes, absolutely. So I didn't mean to imply this notion of functional targeting could actually span networks that span the entire brain, and that's appealing because if you're using non-invasive brain stimulation like tDCS that affects the whole brain, or TMS that affects a target but through indirect effects affects the whole brain, yes, now you're affecting a wide distributed network. I think the same kind of thinking would translate. Thank you.
So let me ask you a question that leads to the link between models and actual studies like the ones many people here are doing. And to some extent it's follow-up of Hardwick's question, which is it's very clear that many studies being done right now on humans are based or as rationale on models. However, models are not biology, and in a way models exist in an interactive way with biology. So my question to you is how do the biological results that people are finding all around, some of them contradictory. Go back to your models, and how do you rebuild your models to address or explain those differences to the extent you can?
Yeah, it's a very good question. Some of this almost gets to some philosophical approaches of how you approach this. But let's say you look at the example of, let's say, BDNF, and you say, well, BDNF is the key. Then now really what we want to use is we want to use animal models to predict what forms of stimulation would best promote BDNF-dependent plasticity. And we could perhaps figure out that the best way to do it is to apply stimulation first, or stimulation afterwards, stimulation of these form of protocols. And so certainly we need to look, the gold standard is the clinical neurophysiologies, or the clinical results. We need to look at them for hints of how to inform the model. But also, you can't try everything clinically, right? You can't sweep 100 durations, 1,000 intensities. People are moving in that direction. You can't try every possible substrate of ongoing activity. Maybe animal models can do that. So you start with a few data points, you parameterize the animal model. Now you make predictions. It may be wrong, it may be right. I think it's fine either way, right? You learn and you re-optimize the animal models to hopefully help the clinical work converge faster. I mean, that's really the goal.
We have a very simple paradox, which is MEP development under anodal stimulation, increase at rest and decrease under cathodal stimulation. However, during motor tasks we have completely different effects, so this would have to be explained by the model.
A lot would have to, yes. I think, we've certainly heard today, especially in Michael Nietzsche's talk and John Rothwell's talk, the complexity, right? So the more people are digging into this issue at the human level, the more they're finding out that anodal isn't just simply excitatory, cathodal is not simply inhibitory, and that there's a huge dependence of the ongoing functional activity. And to some extent, those questions may be able to be teased out at the clinical level, but yes, I hope that these kind of models may help inform that. At this point, I'm just presenting a framework. We really have very limited number of specific predictions that could be addressing the kind of questions you bring up. I guess my sort of hypothesis was just that I think these form of models can help address the exact issues that people are struggling with in the clinical domain.
Very briefly, if you also increase the firing rates of inhibitory neurons at the same time, so how would that affect your overall network output?
So this notion of excitatory versus inhibitory is a very interesting one. So as soon as you have a network and the excitatory neurons are coupled to the inhibitory neurons, it becomes very difficult to talk about stimulation affecting one versus affecting the other, because it's a very tightly coupled network, and the overall state of that network is this dynamical system. So even if the initial stimulation, be it tDCS or TMS, is affecting an excitatory pathway, you're going to see a simultaneous change in the inhibitory pathway. That's why when one tends to go up, the other one tends to go up. And certainly that will affect the kind of plasticity that you see. How is the more complicated question.
So I think we have to move on. Walter?